What we're going to discuss now in this second part of this lecture is the structure and function of nucleic acids. So first we'll talk about the structure of the building blocks, the monomers that make up nucleic acids, so DNA and RNA, which are nucleotides. And then we'll talk about the overall structure of nucleic acids and the four-level primary, secondary, tertiary, and quaternary structure that you can find in nucleic acids. So what are we talking about here? These are the two types of nucleic acid. Again, you should know this from Bio 1, but it's good to review it and add some information as well. The first one is DNA. Please do not use the word DNA. If I ever ask you anything about this molecule, use the full name, which corresponds to deoxyribonucleic acid. This is, as the name says, a polymer of nucleotides called the deoxyribonucleotides. You have four types present in living organisms, and the function of DNA is, as we discussed before, is to store genetic information ultimately. When it comes to RNA, the actual name, which I want you to remember, is ribonucleic acid. So RNA is just the short form for ribonucleic acid. And in this case, differently from the deoxyribonucleic acid, this is a polymer of ribonucleotides. Other four types which are different from the deoxyribonucleotides. They're not interchangeable. I hope you remember that, again, from Bio 1. And then the ultimate function of RNA is in gene expression. You know how it works as an intermediate between genes and the ribosome in the form of messenger RNA, how it helps with the composition also of ribosome in the form of ribosomal RNA, and how it carries, for example, amino acid in the form of transfer RNA or tRNA. And there are other types of RNA present in the living organisms, such as, for example, microRNA short sequences, which again have a key role in controlling gene expression. So let's start with the building block of nucleic acid, which are nucleotides. Nucleic acids, just nucleotides, if you remember, they're made by three key components, which are a pentose, which is different in the deoxyribonucleotides, the one that make DNA, compared to ribonucleotide. So there is a pentose, which is a form of ribose, which can be missing a hydroxy group or not. Then there is a nitrogenous base, always attached to carbon 1' here of the sugar. And then you have one or more phosphoric acid residues, which basically will be phosphate, except for the last one. Starting from the pentose. So this sugar, as I said before, is basically ribose, always in the beta form, which is dictated by this hydroxy group here found on the carbon 1' here, which can be the usual ribose here. In this case, we're talking about, that's the pentose. We talk about ribose, and that's the pentose that is found in ribonucleotides, so those nucleotides that make ribonucleic acid, or RNA. Or might be missing this hydroxy group on carbon 2'. In this case, we're talking about deoxyribose, okay, which is the pentose which is found in nucleotides called deoxyribonucleotides, which are the nucleotides, which are found in deoxyribonucleic acid, or DNA. Please notice that even if you have the same nitrogenous base attached here in carbon 1' of ribose or deoxyribose, you're still talking about different nucleotides because the pentose will be different. Now, one thing that sometimes we overlook is the numbering and naming of the carbons in the pentose. When we look about the pentose, it's important to remember that the carbons are named as 1, so they have a number and then a prime symbol. And the reason why you need to put a prime symbol is to distinguish the carbons in the pentose from the carbons that are found in the nitrogenous base. For example, this is carbon 4. If you call this carbon 4,2, you don't really know which carbon you're referring to. If I tell you, oh, carbon 4 is the one that it's attached to, blah, blah. Whereas if you call this carbon 4 prime, so all the carbons in the pentose as a number and the prime symbol, you know exactly what I'm talking about. So if I tell you carbon 4, now you know I'm talking about this carbon here. If I say carbon 4 prime, you know I'm talking about the carbon present in the pentose. Now, the nitrogenous base is always attached to carbon 1 prime of the pentose, whether it's ribose or deoxyribose, it's always attached there. Now, if you recall from biology, from Bio 1, there are 5 different types of nitrogenous bases, with 4 of them found in RNA and 4 in DNA. All of these 5 nitrogenous bases, which can be called nitrogen containing bases or nucleobases, there are different names used out there. These are the essential bases. They are involved, first of all, on the binding of the 2 strands of DNA, which each other in the double helix, and also they specify the genetic code, so the information that is contained in gene to form the proteins. Now, in general, there are 2 types, genetic groups, which are referred to as pyrimidines and purines. So these 5 nitrogenous bases can be divided into pyrimidines and purines. With pyrimidines, including the nitrogenous bases, which are made by a single ring, and purines, the ones made by 2 rings. So among pyrimidines, we have 3 different types: cytosine, which is found both in ribo and deoxyribonucleotides, and thymine, which is found only in deoxyribonucleotide and therefore only in DNA, and uracil, which is found only in ribonucleotides and therefore ribonucleic acids. And you can see how the difference between thymine and uracil is just ultimately in the presence of this methyl group right here. And in fact, it's thought that thymine has evolved from uracil over time. When it comes to purine, instead, again, these are 2-ring type of nitrogenous bases, and these, the 2 that are included in this group, are adenine and guanine, with both of them found both in ribonucleotides and deoxyribonucleotides. Now, the composition of the pentose and the nitrogenous base is referred to, when they're combined together, as a nucleoside, not nucleotide. Notice the difference in this S right here. So this will be a nucleoside. Okay, they're joined through a glycosidic bond here between carbon 1 prime here and the nitrogenous, the nitrogen here in the nitrogenous base. Now, forget about the information right there, already gave it to you. Now, so nucleotides, different from nucleosides, have additional phosphate or phosphoric groups attached to the nucleoside. So basically, they can be defined as a nucleoside with 1 or more phosphate group. Depending on the number of phosphate groups you have there, a nucleotide could be a nucleoside monophosphate, or if it has 2 phosphate groups there, so a phosphate group, this will be a phosphoric group, a nucleoside diphosphate, or if it has 2 phosphate group and a phosphoric group at the end, so 3 phosphate group here, could be a nucleoside triphosphate ultimately. But basically, the difference between nucleoside and nucleotides is in the presence or absence of this phosphate group. Now let's move from nucleotides, so these monomers that make up the nucleic acid, to the structure of nucleic acids. Just reviewing here. Now, there are 4 levels of structure, similar to what you find in proteins. There are 4 levels of structure that can be identified in nucleic acid as well. We have a primary structure, which corresponds to the simple sequence of the nucleotides, a linear sequence of nucleotides in what we call a nucleic acid strand here. Then, as you know, you have 2 strands that can be joined together, forming a helix there, and this will be referred to as the secondary structure of nucleic acid. Notice that this can be found not only in DNA but also in some type of RNA site, such as tRNA. Then you have a tertiary structure, and this is formed from further bending, if you want, of the secondary structure, which is referred to as supercoiling. And then finally, the nucleic acid can also interact with some proteins. For example, when it comes to histons, you have the nucleic acid are wrapped around histone proteins, or some RNA molecules are found, for example, in the ribosome together with some ribosomal proteins there. These interaction between the nucleic acid and proteins are referred to as the quaternary structure. Let's start with the primary structures. This primary structure is formed by polymerization of the monomers, which are the nucleotides, through the formation of what is called the 3' 5' phosphodiester bond. The 3' and 5' phosphodiester bond comes from the naming of the carbons in the pentas, which are involved in the formation of these basically double ester bond mediated by this phosphate group, hence the name phosphodiester. Two ester bonds mediated by a phosphate group. And so the carbon here, the 3' is the carbon involved in the next nucleotides, if you want. The 5' is in the first nucleotides. Here. Now, this polymerization of nucleotides lead to the formation of what we call a nucleic acid strands. That's the sequence of nucleotides that you can see, for example, here. You have 3 nucleotides joined together. This will be referred to, in this case, this is ribose, so this will be RNA or ribonucleic acid, and so this will be a strand of ribonucleic acids here. Now, these strands, they always have a clear direction, okay, which is referred to as the 5' to 3' direction. If you remember, this is essential to understand how enzymes work. For example, when we talk about DNA polymerase 3 in DNA replication, it works on the 5' to 3' orientation. It gives you information on where actually this enzyme can attach the next nucleotide. By working 5' to 3', it means it can attach the nucleotide only to the 3' here. Going one step back, what do we mean by 5' to 3'? What makes one end 5' and one 3'? Well, it's the last carbon in the pentas that is found at the end of the strand. So in this case, in this strand right here, the last carbon I found at this end of this strand, or ribonucleic acid strand, is the 5' carbon. Therefore, this will be the 5' end of this strand. At this end right here, instead, I find the 3' carbon of the pentas, and therefore this is the 3' of these nucleic acid strands. Now, as I said before, this is important to understand how enzymes work. So if an enzyme works in the 5' to 3' orientation, it means it will get this strand to grow in this direction, 5' from the top to the bottom, meaning that to do that, it should be attaching a nucleotide, it would be attaching a nucleotide only to the 3' end of the strand. So it will be elongating the strand from the 3' end, not from the 5' end. It's a completely different reaction. Okay. Other information regarding strands, in each strand you can find a backbone, which is nothing else than the alternation of the phosphate group, the pentas, phosphate group, pentas, phosphate group, pentas. It's similar to the backbone if you find, if you remember, in proteins. And from the backbone, in proteins you would have the residues, the R groups sticking out. In nucleic acid, you have the nitrogenous base sticking out from the backbone. So these are not part of the backbone. That's the primary structure of nucleic acid. Now, strands, RNA strands, they can stay single strand, or nucleic acid can stay single strand, or they can combine together, forming what we know as a double helix. That structure that was presented for the first time, proposed for the first time by Watson and Crick, 1953. It gave them a Nobel Prize. They got a Nobel Prize for this structure that they came out with, in reality not doing their own experiment, by using information produced by many other scientists, in particular by Chargaffs. They came out with this rule where he observed that if you analyze different DNA samples, if you take, let's say, a sample from a zebrafish or a goldfish, and you take a sample from a chimp, now you analyze the composition in terms of nitrogenous base, you always find that the amount of adenine will always be the same as thymine. So if you have 20% of adenine in the DNA of a chimp, you will automatically know that you would have 20% of thymine. And this percentage will always be the same. If you have 15% in the goldfish, you would have 15% of thymine. Same thing for cytosine and guanine. Same percentage. So if this was 20%, in this case this would be 30%, cytosine would be 30%, the same for guanosine, 30% and 30%. Always equal amount of these two types. But the amount of cytosine and guanine when combined together would not be the same as adenine and thymine. Okay, usually would not be. Just by chance could be the same, but usually would not be the same. And the other thing that I observe is that if you add the percentage of adenine and guanine, it will always be equal to the percentage of cytosine and thymine. And then the other second important information that Watson and Crick used to come out with the double helix structure, it was actually this was the main information to come out with, was the results of the X-ray diffraction pattern that was presented by Franklin and Wilkins. Wilkins was the boss of Franklin, but the experiment was done by Franklin, where she basically took some DNA, like this molecule here, bombarded with X-ray and put a film behind it and see how the X-rays were basically deflected by the DNA. And the pattern observed was very similar to the pattern observed when you look at at helixes in proteins, the secondary structure in proteins. You see a similar pattern, but this pattern was repeated in this case, so meaning that DNA was forming this helix repeated over and over and over. So the combination of Watson and Crick's, of the observation by Chargaffs, Franklin and Wilkins here, and other experiment led to the double helix structure. Double helix then now, based on the model proposed by Watson and Crick, consists of two independent strands when it comes to DNA, or the same strand when it comes to RNA, but in this case Watson and Crick talked about DNA. So talking about DNA, they correspond to two independent strands, so they can actually be separated. They're not joined by covalent bonds, which are oriented in opposite direction. So they are what we call antiparallel. So there are two antiparallel strands of DNA. They are joined together through hydrogen bonds between the nitrogenous bases. So the nitrogenous bases are the bases that forms these bonds that keep the two strands together. And now they do that in a complementary way with having adenine, and this comes from the Chargaff's rule. Adenine would always pair only with thymine, and vice versa, thymine with adenine, forming only two hydrogen bonds, whereas guanine would only pair with cytosine, or cytosine only with guanine, forming in this case three hydrogen bonds. And this is done with these complementary base pairs, which form basically are exactly in the same plane. Okay. And again, this is what I just told you, so that adenine and thymine would always pair only with each other, would be complementary to each other, forming only two hydrogen bonds, whereas guanine and cytosine would form three hydrogen bonds here, only exclusively with each other. Now, this is the structure that was proposed by Watson and Crick for DNA. Similar structure can be found also in RNA, okay, in some RNA molecules, such as tRNA, if you remember, where in that case the big difference is that, first of all, you're not talking about two independent strands of nucleic acid, but it's the same one that folds back and form this hydrogen pairing. The second thing is that in that case you would not have adenine pairing with thymine, but adenine will be pairing with uracil. So take this methyl group out, you would have the pairing that you have in some RNA molecule. So, and usually it's only in some part of the RNA molecule that you would have the formation of this type of double helix. So that's referred to as the secondary structure of nucleic acid. Now, nucleic acid, though, they can stay like that. In that case, you will refer to as relaxed molecule. So DNA in the double helix, just forming the double helix as is, will be referred to as a relaxed DNA. However, in living organisms, you have different enzymes that can cut one of the strands, release it around the other strand, or vice versa, introducing a higher level of structure in the DNA. And this is referred to as supercoiling. If you remember, when you talked about, or when you talk in Bio 2, you talk about DNA replication, and you talk about the first step, the unwinding of DNA. There is DNA that opens up, right, at the origin of replication. This actually is helped by supercoiling. And then you have that these progressively keep opening up, forming the replication forks. And then ahead of the replication forks, you have enzymes referred to as topoisomerase that basically release that stress that will be caused by the continuous separation of these two strands of DNA. Now, this enzyme, topoisomerase, together with other enzymes like DNA gyrases, what it's doing is exactly what I just said. It's cutting, you can see this picture here, it's cutting one of the strands right here and allowing that strand to basically move around the opposite complementary strand, releasing some of that stress. Okay. By doing that, it's actually playing exactly with the tertiary structure of DNA, the supercoiling. Now, this supercoiling again is further coiling for further structure that's formed by this double helix that's wrapping around itself. Here you can see an example of what could happen in a plasmid DNA. Plasmid DNA are small molecules of DNA found within bacteria. They are extra chromosomal, so they're outside. They're not part of the chromosome. They are independent from the chromosome, and they also replicate independently in a similar way than a bacterial chromosome. They have an origin of replication, but they're independent. They're smaller too. Okay, we're talking about 1,000 times smaller than the bacterial chromosome. So these molecules too, this can exist in a relaxed form like this one, but also can also exist in a supercoiled form like this one. Again, due to the action of this enzyme, topoisomerase or DNA gyrases. And now they can form coiling in different ways. What I want you to see is, imagine this as an elastic, okay? And then you can start twisting it, looking from the top to the bottom, and in this case I'm referring to the left side here. You can start twisting it clockwise. Okay, if you do it clockwise, you will be introducing what I refer to negatively supercoils. If you do counterclockwise instead, again looking from the top, you're turning counterclockwise, you are introducing what we refer to as positive supercoils. Okay, so in this case the DNA will be referred to as overwound DNA. If you introduce negative supercoil, you're referring to as underwound DNA here. And if you take a plasmid of DNA, for example, you run it on an agarose gel, something that you should be doing in your lab, and you don't cut this DNA, you can see all of these different levels of supercoiling. The same DNA molecule in this form right here, or in this form right here, would actually, in this form right here, depending on the number of supercoils that you have, would actually go at different speed in an agarose gel. So you could actually visualize it as well if you don't cut it. Instead, if you cut it, it would just become linear, it would lose the tertiary structure, it would be just migrating all at the same level. Okay, but summarizing, the tertiary structure found in nucleic acid, particularly in DNA, is supercoiling. Called supercoiling, it's introduced by the action of this enzyme, topoisomerase, DNA gyrases, and can lead to either negatively supercoiled DNA or positively supercoiled DNA. And all of these has a function. Okay. For example, if you look at the chromosomal DNA in a bacterium, this supercoiling present in the so imagine this being the chromosome in a bacterium. The existence of this supercoiling here, in this case this will be counterclockwise. I see this in front of the other one, so I see it as positively supercoiled. These in this case actually will be, when it comes to really helping one, will be negative supercoiling that helps this. But this supercoiling helps with the first step of DNA replication, which is the opening of the circular DNA molecule. So the opening, the separation of these two strands counteract, if you want, the supercoiling and therefore, at least at the beginning, does not produce too much stress on the remaining molecule DNA. Then when this keeps going, they start introducing instead positive supercoils, which then require the action of topoisomerase. Okay, so in this case, the existence of the molecule with a tertiary structure, supercoiled structure here, helps with the first step. Same thing when it comes to eukaryotes. The existence of this supercoiling in the DNA molecule helps with the formation of nucleosomes. Remember, with DNA wrapping around histone proteins in there. So, and this is also present not only in DNA when it comes to, but also this tertiary structure are found also in RNA molecules, which then later on can use these supercoils also to wrap around other molecules. And so when they interact with different type of molecules, such for example DNA interacting with the histone proteins to form the nucleosomes here, or RNA molecule with a tertiary structure wrapping around proteins, for example in this ribosome here, this interaction here is referred to as the quaternary structure of the nucleic acid. Again, found both in DNA, in RNA proteins as well. Similar to what, in parallel, you would describe as the quaternary structure in proteins, which is when you have two or more tertiary structures, so two or more polypeptide with their tertiary structure, each interacting with each other. For example, in hemoglobin. Same thing here. You have two different type of molecules, so you have your DNA or RNA with their tertiary structure interacting, in this case, with proteins, forming a quaternary structure. So, in summary, what did we talk about in all of this lecture? First, we quickly talk about what biochemistry is. Is that science that ultimately uses the information from many other fields, from biology, organic chemistry, basic chemistry, bioinformatics, molecular biology, to ultimately study the life processes involved in the biomolecules. Then we talked about biomolecules. So we define what they are as polymers of different monomers, which would be, for example, amino acid for proteins or nucleotides for nucleic acids. And we stress on how the sequence of these monomers is essential for the function of the biomolecules. And then we talked about the origin of the biomolecules. First, we talked about the origin of the elements, starting from the Big Bang, flowing up a few billion years ago, all the way to the formation of the biomolecules, explained through these three possible theories: the reducing atmosphere, the deep sea vents, and the extraterrestrial origin of biomolecules. And then, in the last part, we focused on the nucleic acid structure. So we talked about what we're talking about. What are the nucleic acids that we're talking about? So deoxyribonucleic acid and ribonucleic acid. And then we talked about the nucleotide, the deoxyribonucleotides and ribonucleotides. What is their basic structures? How they're made by three basic components: the pentose, the nitrogenous base, and one or more phosphate group forming the nucleotides. And then we talked about how they interact with each other to form first the primary structure through the formation of the 3' 5' phosphodiester bonds, the secondary structure through the interaction of two antiparallel strands forming the double helix. Then we talked about the tertiary structure, how these molecules can form further coiling other than the helix. Okay, and this is necessary for different processes. And then we talked about how the interaction of these supercoiled molecules would ultimately lead to the interaction with some proteins, in some cases like histone proteins or ribosomal proteins, forming what we call quaternary structures.